Welcome, everyone. My name is David Wood and I chair London Futurists. To provide a brief context for this event, let me make a brief personal note. I have been chairing London Futurists, or what became London Futurists, for nearly 14 years now. And before that, there were precursor organisations such as Extra Britannia and the UK Transhumanist Association, whose meetings go back to the beginning of this century. And in these 20 years, we have, on many occasions, addressed the subject of healthy longevity. We've looked at different ideas about bio-rejuvenation technologies, looking at medical aspects, looking at philosophical aspects, looking at economic and political aspects, and so forth. And we're coming back to that subject today with two independent longevity researchers. But what you're going to hear is no mere repetition of what has been said in some of these previous events over these 2 decades. Instead, I believe we are going to be listening to some novel ideas, some breakthrough ideas, ideas which can provide a new clarity to lots of diverse observations and opinions. The 2 speakers are both spent a long time gathering their ideas. Vince Giuliano, who is in his 90s and has been studying this field for at least 20 years, and Steve Buss, who is slightly younger in both chronological age and also time in this area, but he has lots of remarkable insights as well. So I'm not going to say any more now, except that I will ask attendees if they have questions which they would like the panelists in due course to address. The place to type them in is the Q&A window, which you can find below the screen. Otherwise, if you have general remarks, put them in the chat. But as you type, please don't get too distracted, because the main action is going to be taking place on the screen with these 2 remarkable speakers. And with that, I'm now going to hand over to Steve Buss to lead the rest of this event. Steve, over to you.
Well, it's a pleasure to be here today speaking to the London Futurists meetup. And it's also an honor to be here with my colleague and good friend, Vincent Giuliano. Let me show you our agenda for today. We're going to talk for 65 minutes. I'm going to introduce what we're going to talk about in 5 minutes. Then we're going to cover 2 ancient rejuvenation mechanisms for 35 minutes. Then Vince will sum up in 5 minutes. And then we'll talk about practical interventions to trigger these mechanisms. We've got more slides than time, and we're going to move very quickly through this content. The scope of what we're talking about is huge. We're going to give you a 100,000 feet view of the content. We appreciate David Wood giving us the challenge of summarizing what we've learned in a brief time. And with that, we will begin. We've created a draft graphical abstract of what we call the Younging 1.0 explanation of aging and rejuvenation. The key objects around which the Younging 1.0 explanation is configured are aging-related histones, histone methylation, and histone demethylases that interact in a way, and as we understand them, we can see that they both cause and can be leveraged to trigger rejuvenation. These 2 elements have been in almost every life form on our planet, as far as we know, including plants, and most recently, greater leverage over these elements has been established via control of the brain. Vince is going to be talking about histone demethylation rejuvenation, and he's going to be talking about 2 studies that were necessary to pull the content together and to understand it. I'll be talking then about how evolution drove the content of what evolution established in us to establish neuronal control of histone demethylation processes. Then we'll talk about some survival probability benefit examples of leveraging these mechanisms. We'll talk about the puzzle of aging science, and then we'll explain what it would take for us to need to create Younging 2.0. Longevity rejuvenation science is a puzzle of 100,000 pieces, let's say, of a 3-dimensional puzzle. It's difficult to put together for 3 reasons. There's no puzzle box cover with the photo on it of what the puzzle is supposed to look like. Second, the puzzle pieces that you need to put the puzzle together so you can see what aging and rejuvenation is about, the puzzle pieces arrive at different moments. As important, and it's easy to mistake this point, some of the key puzzle pieces that have been needed to understand aging and rejuvenation just arrived within the last 10 years. There's some questions here, but because we want to stay on time, I'm going to shift to the next slide. One of the problems with talking about interventions to lengthen life and to understand aging is that aging theories and theories of rejuvenation often don't completely explain the interventions that actually practically work. For example, near infrared and red light can make a profound difference in health, but most theories of aging and rejuvenation don't address it at all. That's a problem. That tells us that we don't have yet a complete explanation. So at this point, Vince is going to take over and talk about histone demethylase rejuvenation.
Thank you. Some initial thoughts. Longevity science is a little bit in the shape that physics was in the early 19th century. There are many, many different theories. They don't fit together. There are practices that are not explained by the theories. And what's really needed is a new unifying paradigm. And we think we have at least the beginning of such a paradigm here. We think it's very basic. The quest for rejuvenation, of course, is very ancient. I think a very important contextual factor is that there are many natural rejuvenative processes that take place in all species. Things that run the clock a little bit backwards, things like DNA repair, other processes which reset. Based on this, we formulated a concept of Younging 1.0, the whole body version of which suggests that it may be able for us very soon to trigger reverse aging processes that go on for years. That once you've started, it's like aging, but aging in reverse taking place. There is something very basic unifying all of this, which has to do with histone demethylation. And for those of you for which this is a mysterious term, I will get into in a little bit. And it also has a whole genome characteristic That is, it reflects an impact on the whole genome. There is a profound connection to inflammation and aging. As you probably know, almost all degenerative diseases of aging generate chronic inflammation. And finally, we'll get into practical means for generating these age reversal processes and health-inducing processes. So what have we got? What do we think we have? We think we have a framework that is unified by a common molecular mechanism that is common to all natural restorative processes. That is, you look at any of these restorative processes, you're going to find it at work. It underlines a form of younging reverse aging. It relates to and explains most known youth-producing interventions like calorie restriction and hypoxia and infrared light, and a huge list of things that Steve referred to earlier. It applies across species in every biological kingdom, and it's supported by thousands of research studies. So we're making kind of a universal claim for what we have to talk about. We believe that this mechanism is in plain sight, but it's been hiding from the longevity research community. Again, we believe there are thousands of research publications supporting it. We can quote them. I have at least 150 in slides we can show you at the end. It can result in practical, actionable, today, longevity interventions. This is not about a high technology approach coming soon to a clinic near you. It's about something that's available now. And again, it applies to all living species, from single cell organisms to plants to insects to planaria to us humans. And finally, we're going to talk a little bit about how evolution has picked up on this theme and advanced it to bring some of the control of the application of these processes under the aegis of our nervous system so that our nervous system can create it. I believe we're on the road to a grand unified theory for aging. We're on the road to something that would be comparable to what relativity theory and quantum physics did in the early 19th century for bringing together a whole bunch of things in physics under a common framework of understanding. Okay, so I said there are natural restorative processes. Here's a list of some of them. First of all, our epigenetic age, we are all reset to 0 shortly after we're conceived. There's a research switch that happens in embryogenesis very early in the process that resets our age to 0. That's why we can go on for thousands of years as a species, even though our individuals only last 100 years. I'm 92, by the way. I'm planning to keep going till 100. It has to do with the time sequencing of embryonic organ development, renewal of stem cells, which is very important, stem cell fate. As you possibly know, our blood cells are replaced every 15 to 30 days. This is a natural renewal process. If we didn't have that process, we'd never get to 1 year of age. It also explains things like limb and tail regrowth in some species, a list on the right of neurogenesis, DNA repair, wound healing, many, many different healthy responses we know happen in the body, like the unfolded protein response, the UPR. And the key thing is in the lower left-hand corner here, histone demethylation via the expression of a certain protein called JDJM3 plays a key role in each place. And I'll try and lay that out a little bit for you. First of all, there are two kinds of DNA methylation, and it's very easy to get them confused. We have some 30,000 genes arranged in 23 chromosomes. And if there's methylation near the promoter site of any individual gene, in general, that gene is silenced. It's told to shut up. It's told, don't produce your protein. That gene is not transcribed. If you demethylate it, then it is transcribed. And that kind of individual gene methylation is the basis of the aging clocks that Horvath and others have laid out. And it's using this, it is very possible our profile of methylation of our genes change throughout our whole life in such a way that you can actually predict the chronological age of an individual within 1 or 2 years of accuracy this way. And there's been many publications about this. Now, histones are a different thing. There are 23 chromosomes, and in these chromosomes, I once heard that the total length of your DNA, if it was stretched out, would go from coast to coast or possibly around the world. You've got to compact your DNA. And the way the body does this is it wraps them around histones, which can be thought of as spindles or spools. And there are thousands of these, and a single human cell might have tens of thousands. So that and these histones are of a limited number of kinds. There's one particular position in the histone called H3K27ME23. This means histone number 3, position 27, and double or triple methylation. So when you methylate a particular histone position like H3K27ME23, you are silencing the thousands of genes affected by that histone, which repeat throughout all of your genome. If you demethylate that particular histone, you're activating thousands of genes. That's the important difference. And again, a way of talking about this is that when the genes are silenced, and there is a mechanism for silencing this particular histone position called the polychrome repressive complex, the PRC, you have what is called silence chromatin. That is the genes in that vicinity are told, shut up for now, don't express yourself. If you demethylate them, you activate that chromatin. And this protein I talked about, JMJD3, and another closely allied protein called UTX, can activate them. So that there is a natural process that can simultaneously turn on or turn off thousands of genes, is the first point. Certain kinds of methylation, demethylation, having to do with aging, having to do with development, having to do with the intervention, they all are of the histone nature. And included here, for example, and there are publications backing everything we're going to say up. NAD in sirtuins is very popular right now. And histone acetylation, which opens the chromatin up, and methylation, which closes it down, relate to this. NAD has an indirect impact of activating JDJM3. Clotho has been called the magic spinner. It's another intervention. for aging. Again, you need to demethylate the promoter area for the longevity expressions. Hypoxia, oxygen therapy, which we'll get into when we get into practical approaches. Many people take rapamycin, the mTOR pathway. Again, histone methylation plays a key role in all of these. Heterochronic parabiosis. Many of you have heard about this. You sew two mice together, the bloodstreams of the mice, so that they share a common bloodstream. And the old mice gets younger and the young mice gets older by numerous factors. This is research demonstrated effect. Again, this draws on young blood factor, vitamin D3, and numerous health-producing substances, activates JDJM3, and umbilical cord plasma infusions, another approach out there in the longevity community. The viroxia, the variability in the oxygenation of the bloodstream, we believe is the explanation for why umbilical cord plasma infusions seem to convey longevity. In each case, histone demethylation via expression of this one protein, JDJM3, plays a key role. This slide shows the same thing again. This above row here shows normal gene expression, where there's something called the PAL2 complex activated, the genes are expressed. This is closed chromatin gene repression, where these little red mice or little red things show methylation and chromatin complex. There's a number of quotes like the one on the bottom here. These are just things lifted from the literature backing up our points. So let me be clear about what we're claiming and what we're not claiming. We are not claiming, I am not claiming that I began to understand all the epigenetic pathways of rejuvenation. We don't yet. They're devilishly complex. And I actually think that it will take 20 or 30 years before all the various aspects of younging are worked out. We do claim that for most, if not all, naturally rejuvenating processes, JMJD3 activation of histones is necessary, and that it's necessary for most, if not all, longevity conferring interventions. The reason why I say if not all is because I don't know them all. And these are reported observations found in the literature, in the research, drawn from a variety of versions of the biological literature. Some of the lower left-hand list shows some of the areas of research in which you find publication. You find that these processes have to do with why tadpoles can grow new limbs or new tails. It has to do with neurogenesis. You find the literature in dental biology. It's all over the place. And we are also claiming, as shown on the right, we don't understand everything going on. We're like Lewis and Clark, where we think we've discovered a new continent, but there are many, many things to be known about it before it's fairly complete. Steve, for example, just as an example of this, surfaced a publication recently of how it works in heterochronic parabiosis when you sew two mice together. We found a doctoral thesis where he comes out and says, you've got to demethylate H3K27Me. You've got to activate JDJM3 in order to have this happen. there are many, many areas where the literature shows how this plays out. Body cells mature, die, and are replaced by newly differentiated stem cells. Without our repertory of stem cells, which replicate and replace mature cells, we'd never make it to 3 months of age. This renewal process is essential. JDJM3 is essential to it. All stem cells reproduce themselves via normal division. JDJM3 literature shows is necessary for the health and renewal of stem cells themselves. Again, there's a lot of detail here. I won't try and go in it. These are quotes from the literature. Certainly in embryogenesis, the key thing that takes place when we're in a young embryonic stage, our age gets reset to 0. Think of that. It's amazing. Our epigenetic age of every human being is reset to 0. How does this happen? Well, again, our explanation relates to that, and there's literature relating to that. JD3 plays a major role in determination of pluripotent and multipotent stem cells, and many of you are familiar with the literature there. It's necessary to enhance the self-renewal and differentiation capacity of embryonic stem cells and MSCs. So again,
Vince, you asked for a time check. It's now 20 minutes in your section. And I'd also just want to pass on a question from a couple of the audience who are fascinated by your slides, but they hope to be able to get a copy of them later to review them. So, I mean, can you say anything, either you or Steve, about the availability of the slides in their own right, or should people simply go to your standard websites where similar information will be available?
Well, there, in our antiagingfirewalls.com site, we have listed 4 publications, 5 publications that go into detail about this with many literature references. You can go there. This presentation, thanks to David, will be published on YouTube, and you'll be able to pick up the presentation and review it again, or pause on individual slides in the YouTube version of it. So we don't want to hold on to this. We want to let go of this. We want this to reach out. Again, if you look at neurogenesis, our neurons replicate at once. We thought we'd have them for life. We now know they need to replicate. And JDJM3 is necessary. It's part of the step of neurogenesis. And again, I talked about its role in early embryonic development, but this slide is interesting. This is the Marimoto study that Steve was talking about. There are life stages of most organisms, including insects and plants, that we go through that is very early in embryonic development. When our age gets reset to 0, growth and development genes are enabled by JDJM3. At reproductive maturity, in human beings, this would be at around age 20 or 25. Actually, JMJD3 is significantly turned down by the body. And during normal mature aging, our balance of methylation increases of this histone, H3K27ME. And when we get to be advanced aging, it prevails. Later on, we'll talk about how we think there are younging whole body interventions that can elevate JDJM3 and reverse this pattern. So the exciting thing about this is we believe that we are onto the basis of and the practical expression of reverse aging. Again, Vince, what kind of weed are you smoking? This is
I was wondering about that, Vince. As you said it, I thought he must be crazy, but then I realized I believe what you said also.
Yeah, I mean, it's hard for me to fathom how Steve and I could make a breakthrough that people have been aiming at for 4,000 years and never made. And, you know, why me? Why us? And surely this is too good to be true. There's got to be something phony about this. But no, we haven't discovered that. It shows up in research publications, many different categories. Human diseases, it shows up in the research of histones. It shows up in multiple forms of the literature. However, longevity science is one area where it has not shown up, and I can talk about why. There are the publications we've put on our website, and Steve has found one doctoral thesis about the Morimoto, about the heterochronic parabiosis results. But why hasn't this shown up? We believe that there are reasons for this, but we're going to leave those for the discussion theory. Again, there's a very important connection with inflammation. You need to turn off inflammation for JDJM3 in some instances to be turned on. And it serves to turn off inflammation. There's a whole thing, and Steve is going to be talking about one aspect of this called the inflammatory reflex. Here are the titles of the publications we have on our blog. When this slide gets online, you'll be able to click on any of these and go to the publications. Steve, you want to take over here with the inflammatory reflex?
It is unbelievable that all this science, everything that Vince has just talked about, and more, is already in the literature in plain sight. You know, one of the reasons I think that Vince and I have been able to figure some things out is because we're not scientists who do experiments. We just read literature and think about and talk about what the literature is showing, the complete scope of it, and then we try to figure out how it all fits. But let's talk about the inflammatory reflex, which is a different, which is the second major rejuvenation innovation that evolution introduced in mammals. The inflammatory reflex is a label identifying a set of biological objects. In other words, it's an abstraction. It's not a single thing. Kevin Tracy discovered the fact of the existence of a mechanism in mammals he called the inflammatory reflex in the late 1990s. He published a hypothesis paper about it in 2002. A way to think about it goes like this. TIR, the inflammatory reflex, is the label naming a set of mammalian organs, nerves, cell types, receptor types that together perform significant biological functions related to the immune system, inflammation management, and rejuvenation. Despite its importance, the fact of the existence of the inflammatory reflex is unknown to most health professionals. and longevity science enthusiasts. Let's talk about what a reflex is. The image on the left shows a finger that's about to touch a candle. And we all know a hot candle, a lit candle, so that would cause a burn, and the arm would pull away when that happens. Touching the candle and then the reaction to the heat is all regulated with nerves. There is a sensory neuron. It's also called an afferent neuron. A sensory neuron in the end of the finger that would detect the heat of the candle. And if we follow the nerve up into the spine, we see that the sensory neuron interacts with what's called an interneuron, which integrates the fact of the feeling of heat and then has a relationship to a motor neuron, which doesn't connect back up to the finger, but instead connects to muscles in the arm so that one moves the arm away from the heat of the candle. So in the inflammatory reflex, the motor arm of the inflammatory reflex is called the cholinergic anti-inflammatory pathway. This second diagram shows the nerves that are related to the candle situation, the candle context on the left, and then the inflammatory reflex on the right. And because of time, I'm just going to explain what each diagram is intended to mean without reviewing them in detail because there's too much content to review. Here's the interneuron of the inflammatory reflex, the sensory neuron, and then here is the set of biological objects that connect up the interneuron with the spleen. And it turns out that the spleen is a really big deal in the inflammatory reflex for reasons we'll talk about. Here's another view of those same biological objects. We see the brain actually can control the inflammatory reflex through a connection with the spleen through the vagus nerve and the splenic nerve. Vagus nerve. The vagus nerve shouldn't be thought of as a single wire. It's like an undersea cable. There are about 100,000 individual vagus nerve fibers, 80% of them sensory vagus nerve fibers, that is, fibers that in which the signal originates in the periphery and then ends in the brain, and then 20% motor beginning in the brain or in the spine and ending in the periphery. Kevin Tracy once said in a talk that there were about 50 cholinergic anti-inflammatory pathway fibers between the brain and the spleen. Question: why are the vagus nerve and splenic nerve fibers from the brain to the spleen important? They're important because, one, the spleen is part of the blood circulation system. And number two, the spleen is understood to contain about 50% of the inflammatory cytokine cells called macrophages. These cells can exit the spleen and flow to the blood circulation in either of one of two states. Phenotype M1 is an inflammatory cytokine, and phenotype M2 of macrophages is an anti-inflammatory cell. So these diagrams illustrate the scope of the vagus nerve. Here is from...
Can I just mention that JDJM3 is very important in this process. And one of the reasons why it's important is because the expression of it changes the phenotype of these macrophages from the inflammatory to the anti-inflammatory nature. What this is a mechanism that evolution adapted for mammals and for us advanced species for the nervous system to pick up and go beyond and handle automatic functions. Did you know that your nervous system monitors your inflammation and controls it? Well, that's what this is about.
That's right. JMJD3 is implicated in managing the polarity of macrophage phenotype. The metaphor I like about macrophages, I'm doing a little bit of simplification, well, some simplification here, but M1 macrophages, the inflammatory kind of macrophage, I think of them as drunken soldiers who often go on a rampage and not only kill the enemy, but kill their own citizens and women and children and on and on. And the M2 macrophages, I envision the metaphor is they're like UN peacekeepers, right? Who go in and try to heal, establish homeostasis. Macrophages are important for reasons we'll get to in a moment. So there are a lot of diagrams showing the biology and physiology of the inflammatory reflex and the cholinergic anti-inflammatory pathway. We're not going to review any of that. I did want to point out a few things. There is a receptor in the brain called the muscarinic acetylcholine receptor, the M1 type of muscarinic receptor, that is the receptor in the brain that triggers the vagus nerve connection to the spleen. One can trigger it with a muscarinic acetylcholine receptor agonist or an acetylcholine esterase inhibitor. In other words, the way to trigger that muscarinic receptor is by increasing acetylcholine in the brain. It also relates to the autonomic control and management of the heart. This is why heart rate variability, which is a computed, if you think about it, it's not a biological object, it's a computed statistic. The reason high heart rate variability is important and related to health, Kevin Tracy believes, is that it's also managed by acetylcholine. The spleen plays a major role, as I've mentioned before, because it's connected to the blood circulation system, and the macrophages, which manage the fighting of enemies to the host, and also maintaining homeostasis. 50% of them are in the spleen. The reason I wanted to show this second diagram is to show the remarkable fact that not only can the muscarinic acetylcholine receptor be triggered by in pharmacological or supplement means, but it can also be triggered via the Hygge acupoint. The Hygge acupoint historically is among the most potent of acupoints identified in ancient East Asia. That's a profound thing, and I can recount this, my own experience with that, where I have proven to myself that triggering the inflammatory reflex through the Hygge acupoint actually takes place. Here's the spleen. The reason for showing this diagram on the right, in all the diagrams of the cholinergic anti-inflammatory pathway that Kevin Tracy has published about, he never shows that the blood circulation flows through the spleen. And that's the main point, if you stop to think about it, with all the focus today on heterochronic parabiosis and the other major convoy studies where the blood circulation is clearly an incredibly powerful source for both aging and rejuvenation, depending on the characteristics of particles in the blood circulation. I put it to you, evolution has been doing billions of experiments that manipulate the blood circulation such that the way that the spleen interacts with the blood circulation is important. Evolution, I'm going to anthropomorphize here for a moment. Mother Nature thought the blood circulation is important too, and that's why the inflammatory reflex is important. The cholinergic anti-inflammatory pathway, the science around it is complex, just like with histones and histone demethylases, but it's not obscure. Hundreds of scientists and hundreds of studies have worked to understand it. If you go to Google and do a Google search for the cholinergic anti-inflammatory pathway, go to images, you will see all kinds of images from scientific studies giving various kinds of views of how the inflammatory reflex can be depicted. Question: the cholinergic anti-inflammatory pathway, is it positively potent for healthy longevity? Well, the answer is yes. The survival probability findings in 5 studies, 3 about poisoned rodents, 2 about elderly humans using different intervention techniques, are all best explained by the cholinergic anti-inflammatory pathway. And I see I'm running late on my time, so I'm not going to explain this diagram. I have explained it in other places on longevity.org. Here are 2 studies that profoundly show how profound the impact of triggering the cholinergic anti-inflammatory pathway can be if you trigger the mechanism either through the hookah acupoint or through pharmacological means, and then administer lethal endotoxin. Most of the rodents survive. If you don't provide triggering of the cholinergic anti-inflammatory pathway, most of the rodents die. Does the cholinergic anti-inflammatory pathway exist in humans? It's all well and good to have shown that last slide that it does exist in rodents and does have an effect. Is it in humans also? Well, absolutely it is. In a 2014 study, an acetylcholine esterase inhibitor, that is galantamine, which, by the way, I buy on Amazon all the time and I take every day. There was a study done to see the effect of galantamine on the progression of Alzheimer's disease. And after 2 years, the people running the study, when they looked at the numbers, they realized, oh my gosh, the elderly patients who were getting galantamine had survived in such greater numbers as depicted in this survival probability diagram that they stopped the study for ethical reasons. So, yes, ways of increasing acetylcholine in the brain, hitting the hookah acupoint. The mechanism exists in humans, it can be triggered, and it's important. This slide, I'm going to skip talking about it because we don't have time, but I have described in text what's going on in the diagram on the left. Essentially, same point. If you cut the vagus nerve, so the cholinergic anti-inflammatory pathway is effectively disabled, the rodents die faster than they would if you didn't cut the vagus nerve. But in the cases where the vagus nerve was triggered, there is dramatically more survival going on. And what does it mean? Well, it means that there's a mechanism in us that we can trigger by means that we now understand a lot about, and it will increase survival probability. So I was going to say, it's a blank screen. We know that JMJD3 manages macrophage to polarity, right? The existence and presence and number of UN peacekeeper homeostasis concerned soldiers. We also know from another study that the inflammatory reflex triggering the cholinergic anti-inflammatory pathway, it increases the ratio of M2 macrophages divided by the number of M1 macrophages. In other words, these two ancient mechanisms, the histone demethylases, which have existed for hundreds of millions of years, are concerned with macrophage phenotype, and so is the inflammatory reflex. An amazing coincidence, seems to me, it seems to us that Mother Nature has, it's an interesting coincidence. I'm not sure we understand how to characterize it with words at this point, so let's screen blank. Here's the graphical abstract that Vince will now talk about.
Well, again, if Steve reviewed this earlier, we have surfaced a mechanism involving histone demethylation and methylation for turning on very large numbers of genes, which has to do with development. It has to do with aging, and it has to do with younging processes, with processes of restoration. The cholinergic pathway is an advanced form of this. Again, Steve talked about the puzzle. Let's just go on. Let me shall I pick up the slides with my?
Right, let me just say one thing about square, the box labeled B. This 2015 Morimoto study showed that at time of procreation, JMJD3 expression declined, H3K27Me increased, and that caused aging of the C. elegans worms. This diagram to the right, let me find there's my cursor. This diagram to the right shows that in the mitochondria, if the electron transport chain is perturbed, that's the word that's used in the scientific literature, then that increases JMJD3. Wanted to point that out. This figure here is about macrophage phenotype 2 polarity that JMJD3 controls. This diagram here in the lower right of the C box is about the study showing that the cholinergic anti-inflammatory pathway also is concerned about macrophage phenotype 2.
Steve, let me just interrupt for a minute. We're an hour into the presentation.
Yeah. Oh, go.
Sure that many of our viewers are interested in what are the practical implications.
Yes.
What can you do with this? What can we do to apply to generate younging? I think we ought to get onto that.
Yeah, absolutely.
Yeah. So do you want me to take over, or?
Yeah, absolutely.
Okay. What are the things that increase JMJD3 expression? Well, a number of substances like DHEA and its presence in food, a number of natural polyphenols and dietary substances like curcumin, boswellia would be another one. You can activate it by triggering the electron transport chain, and there are compounds. So there's a lot of ways, and I just want to mention that it's important to differentiate between local expression of JMJD3. If you get a mosquito bite or a scratch in your hand and you have a little bit of a wound healing, it's a very local process. And whole body processes, which when we talk about younging, we're basically talking about whole body processes. So all of these interventions are possible ones, and compounds are possible. Now, of these, we've got again, the whole body ones are ones that involve either the dietary system or the bloodstream that ultimately get the message out to large numbers of cells. And we think that the most promising interventions are ones that involve what we call varoxia, or variation in the oxygen supply. This is a slide that Steve was referring to about perturbations in the electron transfer chain. And how can you perturbate the electron transfer chain? Well, in mitochondria, tons of things you can do. You can take, it's a mito-Q, the substance. You can take various antioxidants which have mitochondrial impacts. You can impact through your NAG channel. Through that, you can activate sirtuins, which in turn activate mitochondrial proteins, which affect this, so that a very large category of interventions can be via the mitochondria. Do we know which ones are best? No, we don't. We do know that these interventions listed here all involve and are all fairly good ones. The umbilical cord plasma infusion. Umbilical cords are cut off during the process of normal giving birth. Instead of discharging them, you can save them. The blood in the umbilical cord is going to go into an acute lack of oxygen state. Once you cut it off from the mother, it's going to upgrade hypoxia. Again, calorie restriction is a approach. So a lot of the old approaches to longevity fit in with our explanations, what we're saying. This shows how calorie restriction applies across yield, multiple species. Now, hypoxia and varioxia. How do you express that? Well, one traditional way is just a cuff that you can use on your arm that restricts blood supply, and it's a well-known intervention in the hormesis literature that this has numerous health effects that can improve. If you restrict your blood supply for half an hour in your arm with a blood pressure cuff before an operation, for some operations, you can greatly reduce the possibility of side effects. You can introduce hypoxia and varioxia by extremely high technology approaches like holding your breath. We think that use of a home oxygen concentrator may be an excellent approach. This is a here and now available technology. And there is a guy available here who's got good experience with that technology, and I'll let him talk to that. Okay, Steve, you want to take over?
Varoxia. We're used to the terms hypoxia, which means a lack of oxygen, and a lack of oxygen, if it's not extreme, can be healthy. Also, we know that hyperbaric oxygen therapy is very helpful. That's a paradox. And it was only in 2020 that we learned that there was a paradox, and the paradox has been resolved. We now understand how it works. Let me quickly review this diagram in the lower left. Normoxia is normal oxygen level. Hypoxia is a drop in oxygen level, and that can be healthy. Oxygen level is assessed and evaluated at the cellular level. Every cell evaluates oxygen level because oxygen is used in the electron transport chain in the mitochondria. It turns out that these cells are not concerned, that they don't assess the absolute value of oxygen available to the cell. They evaluate a variance in the oxygen level. So when a cell makes an evaluation about does a hypoxic state exist or not, it's evaluating this drop in the oxygen level. You know, the size of the drop, I'm imagining. But it turns out you can arrange to do to increase hyperoxia in a hyperbaric oxygen chamber, and the drop to normoxia essentially constitutes the cell, because the cell's looking for the variance and the drop in oxygen, that counts as much as the drop from normoxia to an absolute state of hypoxia. And when you get into a state of hyperoxia and then get into a state of normoxia and repeat it, do it intermittently, you're essentially doing what's called intermittent hyperoxia and triggering hypoxic-like healthy responses in cells. This study here's the link here to the study. It was a review of the literature, the hyperoxic-hypoxic paradox. Let's take a look on the far right at the health effects triggered by these two states. In hypoxia, HIF is triggered, VEGF, stem cells, those are healthy things to have happen, but SIRT is not expressed greater, and mitochondria biogenesis is not expressed more highly. But if you do intermittent hyperoxia, then you get the benefit of what varoxia creates. I didn't specify that a friend of mine, Vince Giuliano, I believe he might have been the person to invent the word varoxia, which is the drop in oxygen level assessed by the cell. If you do intermittent hyperoxia, you get a lot more benefit. Let me move to the next slide. So I'm going to tell my personal story here. I use a new therapy type called EWOT, exercise with oxygen therapy. I believe it's the most significant rejuvenation innovation up to this moment. In my opinion, it should be thought of as a required component of any serious rejuvenation regimen. Why? Because it supports doing intermittent hyperoxia in a way that's beneficial. Let me run through how this works. First of all, let me show you my personal setup. I took these pictures this morning of my equipment. I have, an extra aerobic exercise machine. It's a stair step with arm movement. I have an oxygen concentrator here, and I have a bag that I put on my ceiling with just simple hooks because oxygen doesn't weigh anything. Here's a picture of the bag fully inflated. That bag holds 900 liters of oxygen. Here's on the upper right, a close-up of my EWA exercise machine. Here's the mask that I use. I have some BFR bands, blood flow restriction bands. The idea here is that while doing aerobic exercise, you put on a mask and then you use CPAP hoses to connect up with a bag that's holding highly concentrated oxygen, not 100%, but like 90%, 95, 85, depending on how the settings you use to inflate the bag with oxygen. And while you're doing aerobic exercise, you're breathing in the oxygen. I can, when I do EWA, exercise with oxygen therapy, I can blow through, I would say, 800 to 850 liters of oxygen in 16 minutes. To understand why this is so essential, and we don't have time to do this, but I suggest 4 steps to really understand it. First, watch the Robert Rowan YouTube video. And here's the Robert Rowan YouTube video on the right. Then take a look at that hypoxic paradox article that I referenced earlier. The reason EWA is so helpful is because it triggers intermittent hyperoxia. I also would encourage you to take a look at an article that was published in 2019, the microcirculatory theory of aging. Basically, as we age, capillary density decreases dramatically. We have fewer capillaries that are actually functioning in a healthy way to bring oxygen to the cell for purposes of generating energy and providing nutrients to the cell. Finally, step 4. There's a German who published a book in English. It was published in 1990 called Oxygen Multi-Step Therapy. It was written by, here's the book, what it looks like, published by Manfred von Ardenne, remarkable guy, born in 1907, died in 1997. He did a demo of the TV that he invented in the 1930s. He did a demo to Adolf Hitler. After being captured by the Soviets after World War II, he assisted as a lead kind of manager. I don't know what his role was, but the Soviets knew because he was awarded 2 Stalin prizes. Is that a remarkable thing? In 1960, he decided to devote his time to biology, and he created a remarkable protocol for health called Oxygen Multi-Step Therapy. He closes his book with this statement explaining how oxygen multi-step therapy, his findings were that it added 1.5 decades to healthy human life. So my personal experience is that discovering and doing EWA transformed my life in a way I never imagined possible. I now understand how profound the mechanism of action actually is. And here's what's going on with it. Doing EWA triggers intermittent hyperoxia, varoxia, throughout the body, including capillaries that are reopened by EWA because of the blood circulation pressure triggered by aerobic exercise. 2, Viroxia, as we've already seen, perturbs the electron transport chain, which 3 triggers JMJD3, 4 resulting in H3K27Me3 demethylation, the rejuvenation process we call younging. This is my favorite, best approach. The reason I think it's best is because this is the, in the way I think about it, this is the only approach that impacts every cell that the blood circulation system touches. Now, there may be capillaries that are closed or atrophied or whatever, but the pressure of the exercise can open them up, and then those atrophied cells can get oxygen. No study has been done, but I believe it would increase the capillary density that this step 3 note references. So this is my favorite histone demethylation practice. I do some others also. I have, what I'm talking about, I have a near infrared headset that I use. It's great. I can feel the difference when I use it. And that's all that I have to say about this.
Well, that's fascinating. We are now...
We're way over time.
I will let this extend further because there's a whole bunch of questions that have come in, and I would like to try and answer at least half of them before we break. Is there any concluding remarks that either of you would like to make before we turn to some of these questions?
Just the fact that this is an ongoing story. We're telling a first chapter in this story. There will be many, many other chapters and many other things. The nature of the story is interesting because once we got into this, we surfaced more and more that's relevant. And we've covered some of that, but there's more and more to say. And for those of you who might be interested in this and who have the skill and background, I suggest that you explore it on your own and help chime in with the development of it. And I agree, we should go to questions.
So I will take questions now. I'll take questions generally that have had the most thumbs up votes, and most of the audience haven't been voting yet. So if you do see a question there you would like to be prioritized, put your pointer on it and raise it. I'll start with a question about safety from Martin Berlet, who says, you've talked about some positive impact of JMJD3. Are you sure you know which genes are affected by it, and might there be some bad side effects?
There is much to be learned. Much that we don't know. And there is some literature that JDJM3 is a process hijacked by cancer cells in the interest. In fact, all of the growth and development processes that we encounter, like IGF-1 and many others, are subject to the fact that cancer cells can hijack them and use them. We believe that if you create a healthy anti-inflammatory body process in your body and you limit your chronic inflammation, that there's probably no major lists of cancer. But other negative effects may well appear. Other positive effects are likely to appear.
Another question about safety from Adrian Cole. How can you be sure that the histone demethylation reprogramming won't turn on genes that actually are not required in a particular cell? We talk about reprogramming as making the cell younger, but won't it possibly also have side effects too?
There are so many genes and so many side effects that we simply don't know now. I cannot be sure. And this is the nature of science. You can never be sure. And do I know of any? The only negative things I've seen have been in conjunction with cancers.
If I could just say, chime in here too about that, the processes, many processes, many interventions that the longevity science community has known to be helpful for a very long time: fasting, hyperoxia, DHEA, and the others on the list that Vince has listed. There's the sirtuins, NAD. They are all involved in JMJD3 related processes. Vince is right, absolutely, that JMJD3 is associated with some cancers. What we wonder if JMJD3 doesn't in some ways, quote, know that cancer is a greater risk, and that's why it reduces its expression at a young age.
Yeah.
Let's take a question about skin from Chris Wickman. He says he's in his late middle age chronologically, and many of his aging markers seem to be around 25 to 35, but his skin seems to be aging much faster. Does your theory of younging have any potential to reverse skin aging? And is that actually important as well?
The answer is yes. I do not understand all the aspects of it, but definitely yes. There are definitely yes, and again, there is incredible complexity underneath what we said. I've tried to simplify it out, most of the complexity, but there's incredible complexity.
So I've looked at this. Retinol, which is has been shown to increase youthfulness of skin, is a JMJD3 promoter. Also, near infrared light and red light has been shown to increase youthfulness, the youthfulness of how skin looks. Near infrared light and red light are also JMJD3 promoters through the electron transport chain mechanism.
A question from Tony Zarneski. He asks about drugs other than just rapamycin that might have effects in histone demethylation or on the inflammatory reflex.
Well, one major drug is metformin, which is used by many people as a longevity intervention. I'm aware of many people who are on metformin, not for dietary reasons, but simply because of its, and this is clearly an example of perturbation of stage 1 of the electron transfer chain. It fits right into our theory.
So do either of you want to share which supplements or drugs you're taking in addition to the oxygen mechanisms?
I am taking approximately 120 pills a day, and the main a major supplement I depend on is one which we developed ourselves for control of chronic inflammation. Again, for many of the magic effects to happen, if they cannot happen in a body which is in a hyperinflammatory state. So I would refer you to some of our blog entries for particular supplements and drugs, and there's a long list and a long list of reasons and a long list of arguments.
There's a question about the spleen. Deborah Colo asks, what happens to people whose spleen has been removed via splenectomy? Do other parts of the body, such as the liver, take over that function, or are these people doomed to not benefit in the same way?
That function does not hold only in the spleen. It holds in other parts of the body. The spleen bears a major responsibility for it.
And is there a possibility of having an artificial spleen, or some of the functions could be taken over by other means?
Well, as I said, in the normal body, the cholegenic pathway that Steve described, the spleen is not the only one that produces acetylcholine and the good effects. But I think what are the implications? It's probably a good idea to keep your spleen if you have it.
Right. About substances to take, there are two ancient mechanisms. Let me do a quick segue before I talk about practical interventions. We think at this moment, we could change our minds tomorrow, that there are only these two ancient mechanisms. We don't see any evidence that there is a significant other ancient mechanism for rejuvenation other than these two. And so many times we know of an intervention that's healthy. The first thing we do is we go do a Google search and see if that intervention trigger is related to one of the two mechanisms. So far, we've never come up short finding that it's related to one or the other or both. So I think we're developing our own personal regimens around these ideas, ensuring that you've got enough acetylcholine related levels in your body is really important for triggering the cholinergic anti-inflammatory pathway. By the way, REM sleep is controlled by the same receptor, that M1 muscarinic receptor that drives the brain to spleen mechanism.
And one mechanism.
Galantamine has already been shown in human studies to trigger increased survival probability. It's sold on Amazon and on other sites.
There's a question from Stephen Miller about your research in the literature in JDJM3. How does this fit with the work of Dr. Harold Katcher into mechanisms for rejuvenation by a fraction of young blood plasma?
We think it fits very well. He has a fraction of young blood plasma that is proprietary to himself. We don't know what it is, but he has published Jungian results that show that for a variety of small animals, for a number of small animals, tracked over a 2.5-year period, they go into a reverse aging process. Younging actually happens for those animals. You can look at the indicators of their ability to handle a water maze, their inflammatory indices. You can look at numerous indices in papers he's published. And so that the very existence of younging is demonstrated by Katcher's work. I have a lot of respect for Harold.
Steve, a question about your O2 machine from Paul Bolshemin. Did you build it yourself? Could a machine like this become commercially available for others who would like to try it as well?
It's commercially available. This kind of system is available from multiple different companies. You can buy a complete system from multiple vendors. You can buy the concentrator. So let me list the components. Concentrator, various hoses, the hose from the oxygen concentrator to the oxygen bag, and the CPAP hoses from the oxygen bag to the mask. You can buy everything from one vendor. I actually built my own bag the first time. The only reason I gave it up is because it leaked too much, right? But so then I bought a new bag. If I was making any suggestions, I would get a mask that had two adapters to take CPAP poses. You can get those at optimalbreathing.com. And you can also get the bag to support two inputs to the oxygen mask. There's also a company called Live02, which is probably the most well-known. In terms of building it yourself, I don't know that you could build an oxygen concentrator. I guess if you had the skills to do that, you could do it.
So just talk about where's the best place to get oxygen. Does it matter what kind of oxygen? Martin Bielecki asks if there's a difference of the kind of oxygen that could be used in here in terms of pure versus impure.
Well, it turns out that the oxygen generated by the oxygen concentrator is at a level of 90% to 95% oxygen. Of course, a hyperbaric oxygen chamber is 100. I haven't read this, but I assume it's 100% with pressure. But to get the benefit of exercise with oxygen, you don't need it to be a 100% pure. More important than pure, getting 100% pure is the amount of time, the number of sessions that you can spend doing it over the course of 6 to 8 weeks, 15-minute sessions a day, as outlined in the Von Arden book. The Von Arden book has a bunch of protocols. Doing it consistently is more important than the absolute purity. And buying oxygen tanks is a lot more expensive to do over time than buying a concentrator that is basically, once you pay for the concentrator, the oxygen is free.
We will transition in a minute to a more open format in which people who've had their hands up or whose questions haven't been reached or who have been too shy to ask questions will be able to enter the discussion as well. But I would like to try and push in one or two more questions before we make that transition. I'd like to ask about the philosophy of aging here, or the theory of aging. How might it be that nature has provided these mechanisms but hasn't actually taken advantage of them? Or maybe some of the creatures that have negligible senescence are taking advantage of these mechanisms. So why do most creatures still grow old despite these mechanisms being deep in nature?
I think, first of all, nature is very flexible and evolution is flexible. If the last 130 years have seen a doubling of the average human lifespan, and our increase in height by 6 inches as human beings, evolution is very much alive and active today. I think if you look at evolution, you have to couple in social evolution with biological evolution. Social evolution is things like clean water, clean air, seat belts, all the things that public health measures that we've taken that have helped and contributed significantly to our average lifespans. So I see what we're doing in this presentation, and as scientists, is seeking to contribute to social evolution that ultimately will facilitate biological evolution. Why has nature decided? I think nature made a very early decision, which is well known. It says, pay attention to the species, not the individual. Maximize life for the species, and there have been many, many theories that the nature doesn't give a damn about a species after you reach reproductive age. And that is consistent with what we see, that, after the age it takes for you to raise your children, the DNA methylation via the protein we've been talking about goes on. And So, but this is a whole other discussion. I'd be glad I take it on you.
So 2 quick points about that. First, there are species with extremely long lives that are pretty famous, right? The naked mole rat. Some species of bats, whales, turtles. Someone challenged me about turtles recently. I looked it up, did a Google search. Turtles experience hypoxia. Naked mole rats, they live in filth, they experience hypoxia. The long-lived species of
They live
bats.
in tunnels underground where there's
No
different.
oxygen, that's right. In other words, in other species, hypoxia and the habitat, right? Their living context habitat is hypoxic. Secondly, about the question, why hasn't evolution taken more, really helped us to use these innate processes to date as much as what is potential? There's an assumption underlying that question, and the assumption is this, that seems like evolution should have had enough time by now to optimize human understanding and use of these mechanisms. Clearly, there hasn't been enough time, but these mechanisms can be consciously triggered. That's the exciting thing for humans, that we can understand them and begin to try to trigger them.
The argument I've sometimes heard is that we have biology which makes sense for the environment in which we found ourselves. But if we can change the environment, then we have more ability to change our biology, which will give us the benefits without having the drawbacks.
Yeah.
Certainly, major discoveries like we discovered about germs, we discovered about bacteria. Prior to bacteria, if you had an infectious disease, you saw a priest because they were the only experts in this. And we hope that pursuing this line further as it unfolds, there will be many things that open up.
My final question, which I'll give you both as a chance to make concluding remarks, is why haven't more of the longevity community, the other researchers, paid more attention to these mechanisms? And are they starting to pay more attention based on the preliminary publications that you have been making?
Well, as I said, there are thousands of publications relating to JDJM3 and this histone methylation, and they are specific to all kinds of areas of literature. It's as if research takes place in finite communities. This has been studied. The history of science has been studied, and it's known that a community of about 200 people studies a topic. Their graduate students refine what's done. They dig deeper. The reviewers are part of the same community. They don't talk to anybody else necessarily. There's about 1.5 million publications in PubMed in the biological and health sciences literature every year. Nobody can begin to approach these. So that our entire structured apparatus of research is oriented towards dig deeper, find out more. And we're thankful because a lot of people have dug deep in the community. Now, why has nobody picked this up earlier? I don't know. I think it's, nobody's paid to do that. Everybody's paid to do the research that their research grant says, and they need to renew their research grant. And their research, their graduate students work on the same research grant, and their survival depends on deepening their own body of research. So that I think the whole structure of research has to do with the fact that if you want to know what's going on and you want to discover things, research in the literature, we're beyond the point. Get a lab, get a computer, become a master at search engines, become a master of that. That's what Steve and I have done.
Thanks. Steve, any final comments from you?
I agree with everything Vince said. I'll just add one last thing, that we don't do experiments. We're interested in the science, and all we do is read broadly. And I think there's not a lot of people who are as crazy and obsessive as
yeah. And we're not
Vince
paying. We're not tied into a grant that means go deeper and deeper into the area of your grant.
Yeah.
Well, in a moment, I'm going to take a break. We may break for about 8 or 10 minutes to allow people to have a comfort break, to recharge coffee cups, make a investment bids on your smartphones, whatever you want to do. Before that, I will say a couple of things, or rather, I will read out a couple of comments from the chat. Stephen Miller said, "This is a ridiculously crazy and exciting presentation. What could be more important?" Walter Crompton says, "Best presentation I have seen in a long time, maybe ever." Tony Zarneski says, "This has been a truly profound presentation on a better understanding of the underlying processes of aging and the potential regeneration of a human body." And there's lots more, but that's representative examples. I just want to add my thanks to Vincent and Steve for boldly setting out. Cutting edge research is by no means finished. People can surely, if they want, snipe at it and pick holes in it. But the point is, you are inviting people to become more involved in this conversation, which I will do shortly too. So if people are still around in about 8 or 10 minutes, I will give everybody a chance to transform from being an audience member into a panelist, in which case everybody will have the chance to turn on their cameras if they wish, to turn on their mics, and I will give people maybe up to 90 seconds initially to offer remarks. We'll take remarks in batches, and then Steve and Vincent can reply. And we'll do that for maybe 30 or 40 minutes. Before that happens, I just warn you that if you accept that invitation, you will lose everything in the Q&A window. So if there's something there you want to copy out, you'd better do that before accepting this invitation. So thanks once again, and I'm now going to switch off the recording and wish everybody a very healthy and long future, a future of lots of younging. Thank you.
Thank you, Dave.